Here's an overview of what we're going to cover. First of all, I have 6 definitions that I think you should know. First of all, spectroscopy/spectrometry is a study of the interaction of energy with matter. Chromatography is used to separate and analyze mixtures and can be paired with other techniques for further analysis. Stationary phase is the phase which separates the analyte to be measured from other molecules in the mixture based on differential partitioning between the mobile and stationary phases. Chemical affinity is the property by which the dissimilar chemical species, for example atoms, molecules, and ions, are capable of forming chemical compounds. A polymer is a large molecule composed of repeating structural units. Retention time is the time it takes for an analyte to pass through a system under set conditions. I'll also be covering 2 chromatographic techniques in this video. The first one is gas chromatography, mass spectrometry, and I'll also be covering high-performance liquid chromatography. All forms of chromatography rely on a mobile and stationary phase to separate compounds. Now you may ask, how do scientists find out about the dangerous elements and compounds in the environment? They collect samples of the elements and compounds and then inject it into a GCMS. Gas chromatography is often coupled with mass spectrometry in a technique called GCMS analysis. The gas chromatograph separates components of a mixture, while the mass spectrometer then gives structural information about each one. GCMS can also provide quantitative data when standards of known concentration are used with the unknown. In GC analysis, a minute amount of a mixture to be analyzed, typically 0.001 milliliters or less of a dilute solution containing the sample, is injected by syringe into a heated port of the gas chromatograph. The sample is then vaporized in the injector port and swept by a flow of inert gas into a capillary column. The capillary column is a thin tube, usually 10 to 30 meters long, but only 0.1 to 0.5 millimeters in diameter. It is contained in the chamber, which is also known as the oven, whose temperature can be varied according to the volatility of the samples being analyzed. The inside of the capillary column is typically coated with the stationary phase of low polarity, essentially a high boiling and very viscous liquid that is often a nonpolar silicon-based polymer. As molecules of the mixture are swept by the inert gas through the column, they travel at different rates according to their boiling points and the degree of affinity for the stationary phase. Materials with higher boiling points and stronger affinity for the stationary phase take longer to pass through the column. Low boiling and nonpolar materials therefore pass through very quickly. The length of time each component takes to travel through the column is called the retention time. The retention time typically ranges from 1 to about 30 minutes, depending on the sample and the specific column used. As each component of the mixture exits the GC column, it travels into a mass spectrometer. Here, molecules of the sample are bombarded by electrons. Ions and fragments of the molecules are formed and a mass spectrum results, similar to those we have studied in Chapter 9 or 10. The important thing, however, is that the mass spectra are obtained for each component of the original mixture that is separated. This ability of GCMS to separate mixtures and give information about the structure of each component makes it a virtually indispensable tool in analytical, forensic, and organic synthesis laboratories.
Here, we will separate a mixture of methanol and methylbenzene. About 0.1 microliters is taken up in a hypodermic syringe. It is then injected into the inlet port through a self-sealing rubber disc called a septum. The first peak is methanol. the more volatile component. Its retention time, that is the time taken for it to pass through the column, is about 1 minute. The second peak, with a retention time of about 1.5 minutes, is the less volatile methylbenzene. Its peak has a larger area, showing that there is more of it in the mixture. The computer calculates accurate retention times and peak areas. The area under each peak is proportional to the amount of each component. The computer calculates this. This instrument is used for undergraduate practical sessions, and samples are run singly as and when each student is ready. In this alternative instrument, the gas chromatograph is on the left, and the detector is a mass spectrometer which runs the mass spectrum of each component as it comes out of the column. This can be particularly useful when analysing an unknown mixture, as the mass spectrum can help to identify each component. The combined technique is called gas chromatography mass spectrometry, GCMS.
High-performance liquid chromatography, also known as HPLC, is another chromatographic and an analytical chemistry technique that can be used to separate and analyze mixtures of chemicals. The HP part of HPLC can stand for high performance or high pressure, since a pump is usually used to push the sample through to the mobile phase and finally where it is analyzed. High-performance liquid chromatography is similar to other chromatographic techniques, but it utilizes liquid in both the stationary and mobile phases. Typically, scientists do some sort of chromatography to separate and then use another method to further analyze the separated compounds, like mass spectroscopy. Basically, whenever there is an unknown mixture, the process it goes through is separation and then identification. Separation is usually done with some sort of chromatography. Afterwards, identification is done by using spectroscopic techniques such as GCMS, library matching, or nuclear magnetic resonance spectroscopy, also known as NMR, and may be followed by formula confirmation by accurate mass measurement. The compounds are separated by having different stationary phases, such as having the sample go through water or methanol or other organic solvents. After the sample is separated, many techniques can be used to analyze it.
In HPLC, the stationary phase is a solid packed into a column, like one of these. This particular column contains silica particles to which C8 hydrocarbons are attached, making the stationary phase nonpolar. In HPLC, the liquid is forced through the column by high-pressure pumps. The whole apparatus looks like this. These bottles contain solvents. Two solvents can be mixed in any proportions to give a mixture, the liquid phase, of suitable polarity for the separation that is being done. In this case, one solvent is water, very polar, and the other ethane nitrile, less polar. The operator can decide on a mixture with the correct polarity for the separation she is doing. These are the pumps. They produce a pressure of 15,000 kilopascals, 150 times that of the atmosphere, hence the name high-pressure liquid chromatography. If a single sample is to be run, it is injected into the solvent stream here, in the injection port, via a hypodermic syringe. Alternatively, several samples can be run in succession by loading them into this auto-sampler, which will run them in order without any human intervention. The pumps force the mixed solvents through the column. The solvent emerging from the column and carrying the separated components of the mixture passes into the detector. Here, a beam of ultraviolet light shines through it. This light is set at a wavelength that is absorbed by all the components to be separated. When the detector reading drops, a component that is absorbing UV light is coming out of the column and passing through the detector. Many alternative types of detector are possible. This one measures refractive index. The time that each component takes to come off the column is called its retention time and can be used to help identify it. Here, the HPLC instrument is being used to separate a mixture of two steroids used in a pharmaceutical preparation. The column chosen is packed with a non-polar solid. The tails of the molecules represent hydrocarbon chains C8H17. Having chosen the solvents, detector wavelength and flow rate, a single sample is run by injecting about 20 microliters into the injection port. The more polar component comes off the column first, followed by the less polar. The peak at retention time, 1.5 minutes, represents other ingredients used in formulating the product. This is the pharmaceutical product, and behind it, its chromatogram.